All right, thank you, Tracy, and thank you, Scott, and the LGS Foundation for having this wonderful event, and also inviting me here to give a talk. Okay, so I'm Jenny Hsieh. I'm from the University of Texas at San Antonio, and today I will talk to you about our unpublished work addressing the role of CHD2 using brain organoids from patients and also from gene editing. So, okay, so we've already heard how it's really that there's a huge genetic basis for DEEs, and really inspired by the work of Gemma Carville and Heather Mefford, who's here today. We decided to take a look at a couple of these monogenetic epilepsies in looking at their roles in development, especially in induced pluripotent stem cell models. And so today I'll talk to you about CHD2 as an example. So CHD2, there'll be a few talks this afternoon on different models, and so I can just briefly summarize. It stands for chromodomain helicase DNA binding protein. It's a mouthful, but generally these chromatin proteins are really important to regulate gene expression. They could be activators and repressors of gene expression and sit as these master regulators. And so there's a bunch of them. And they've CHD4, 5, 7, 8. They're very interesting because they've always been associated with a brain role in the brain, either in normal development or disease. And so not much was known about CHD2. And it's hard to see here, but in this little cartoon down here, there's a bunch of mutations that have been identified in CHD2 that have been linked to epilepsy from different groups. So we became fascinated by that idea. We also started looking at the literature, what is known about CHD2 in neurodevelopment, because we're really interested in which cell types that CHD2 plays a role, how does it affect the brain development. And we found this paper in 2015 by Hsieh et al. They used a hairpin RNA to reduce the level of CHD2 protein expression. And they described in mice early stages of mouse development using different protein markers of progenitor cells. There's a reduction of that marker. This marks like the neural progenitor cells when you reduce the level of CHD2. And there's also, interestingly, an upregulation of markers of neurons. So suggesting that maybe CHD2 plays a role in early stages of brain development. And Bobby Hunt will be giving us a talk on more about the role of CHD2 in mouse models. So Vanessa, a postdoc in the lab, really spearheaded this project in order to try to develop patient or human-specific cell-based models with the idea that there could be differences between a way the mouse brain develops and the way human brain develops. And because it's very difficult to study and to access human brains while they're developing, we really became fascinated by the use of organoid models. So the way we kind of approached this is we made induced pluripotent stem cells from patients, but we can also use human embryonic stem cells and use CRISPR gene editing strategy to remove CHD2 from the genome. But first, what Vanessa did was just interrogate different models of making brain organoids, and she focused on this model that was developed by the Sergio Pasca's lab at the Stanford University. And so in this model, in these sections up here on the top panel, you can see they resemble cortical structures. So at either 1 month or 3 months, you begin to see this beautiful tubular-like regions recapitulating how a brain develops, an infant brain develops in the ventricular zone, and then are surrounding the ventricular zone. You can even see like markers of these early genes that express the progenitor cells. And then with additional time in development in culture, you also start to see markers of neurons, like even cortical layer markers, like the deep layer, and then with additional time in culture, those superficial layer neurons. Of course, you have to continue to grow these in vitro, and it could take months and months to develop those structures. There's also quite a bit of heterogeneity in these structures, and there are many investigators now trying to optimize these in vitro protocols. Vanessa also uses a way of using factors to also recapitulate aspects of the ventral brain development. These structures, these subcortical structures, express markers like GABAergic interneurons or calretinin interneurons. And so using these brain organoids, we can kind of pattern and make both the dorsal and the ventral regions of the brain, at least aspects of this in culture. Okay, so at least I kind of alluded to this. We decided to take these parallel strategies. One case, we tried to recruit patients with CHD2 mutations and make their iPS cells, and then we would try to compare cell-based phenotypes between them and healthy controls, stem cells from healthy controls. Alternatively, you can take a line from a healthy individual, and then you can genetically or edit the genome of CHD2 in order to now introduce the mutation or knock out the gene in the background of the healthy patient line, or vice versa. You can take the patient line and you can recover or restore the gene, and then you would have these isogenic controls. So in the first set of lines, we used CRISPR, and we basically just took a human embryonic stem cell line. This is the H9 cell line that many labs use. And we used CRISPR to randomly generate an insertion or a deletion, and we were able to obtain 2 different mutations that were predicted to cause a loss of function or to knock out CHD2. And then in the other set of lines, we were able to identify families with children with CHD2 mutations, and in this we collected samples from 3 female individuals. 2 of them, turns out they're twins, so they have the same duplication of CHD2 on the same area of the chromosome, and it was predicted to be a loss of function, and from the 2016 paper. So we made iPS cells from these female patients, and then we compared this to a 3rd female patient line that has a deletion on a chromosomal deletion. And then again, these patient-derived lines we tried to match the control lines by healthy in terms of at least do our best to try to match for the age and the gender, but it's really not ideal. Okay. And so one of the ways we started to think about how to study CHD2 in these iPS-derived organoid models is to first identify if the cells that we're generating actually express CHD2, this protein that we're interested in. So at this time, we didn't really have a great antibody to survey and to find out which cell types express CHD2. So what we decided to do was just to perform qRT-PCR to try to detect the expression of CHD2. And so as a positive control, just to see if CHD2 is even expressed in human brain development, this is when we were back at UT Southwestern, and we collected brain tissue, fetal brain tissue, just from different stages, like late 2nd trimester or early 3rd trimester, even late 3rd trimester, et cetera, et cetera. And so it's very difficult to see, but it's just saying we were just looking at, using these human samples over here to just try to benchmark a little bit where we expect CHD2 to be expressed, and then compare that to our organoids, both from the cortical and the subcortical organoids. And what we can see is we can detect CHD2, at least its RNA, mRNA expression in our organoid systems, and it appears to have even more expression as the organoids are grown in culture, where they're starting to produce more neurons. So we get this impression that maybe CHD2 expression is found more prevalent in the neuronal population, although the best thing we should really do is survey this on a single cell level and maybe spatially as well. Okay, so then the next set of data, we wanted to look at just sort of take this reductionist approach to basically try to see if we can explain how a CHD2 mutation would affect development. And so we looked at the growth of the organoids as just sort of a surrogate measure of how their, how their morphology is, how are they growing in vitro. And so again, on the left is our comparison with our CHD2, CRISPR knockout cells, and then on the right is our patient-derived iPS cells. And the top panels are the cortical ones, and the bottom ones are those subpallial ones, the ones that contain these GABAergic progenitors. And basically, we didn't see a huge change in morphology. It looks like when in the CHD2 knockout, they were a little bit smaller at some of the time points, but overall, there was no significant difference. In terms of the patient iPS cells, they appear to be bigger. So it looked different than when you had the knockout. So that's one thing we noticed right off the bat. And to try to explain why the cells from the patient-derived iPS cells are larger at both the cortical and the subpallial, we really will need to go in and kind of look cell by cell to explain why they're larger. So one of the things that we decided to do is kind of take a less biased approach. So what Parul decided to do is use single-cell RNA sequencing, where she could dissociate these organoids into single cells, sequence the RNAs as within this individual cells, and then use a collection of different genes to assign identities of cells. So here on this slide, we're just basically showing you some of the genes we used to assign identities to different cell types, like the radial glial cell. The radial glia cell expresses genes like HES1 and HES5. Interneuron progenitors will express some of these other interneuron progenitor genes shown here. There's also cycling progenitors, et cetera, et cetera. And so we could use this sort of unbiased approach to identify various cell types in our organoid cultures. This is just showing you an example from one control organoid. And so what Parul sees is now you can take a look at, based on these cell identities, these cell clusters, and compare them between our control and our CHD2 patient-derived organoids. And interestingly, the largest difference we see when you compare those populations is an increase of these intermediate neural progenitors and a reduction of deep layer neurons, which again, we're not really sure if this explains that larger, organoid in general. This is a very different readout. One was looking at the size of the organoid. This one is we're dissociating them and measuring mRNA. So again, we're not really sure how it connects, but it's interesting that there's these differences in the population. Okay, so then finally, one of the things we really want to be able to kind of connect this is in terms of how do these changes in cell types and growth properties in the organoid system, does it affect the function of the cells? And so it really kind of, we wanted to measure the physiological properties of organoid-derived neurons, and this area of investigation is definitely more preliminary, and I think in general the field is trying to figure out what's the best way to measure the neurophysiology of these organoids. And so what Zane Librand did is he took advantage of these multi-electrode arrays. So these cell culture plates, at the bottom of the plates there are these different numbers of electrodes, and some people, they grow their organoids directly on the MEA systems. Some people, like what we do, is we grow them separately, and then we use this protocol which developed by the Posca lab, where you could then fuse the cortical and the subcortical structures together to allow those interneuron progenitors to migrate into the cortical side. And then we place them gently on the multi-electrode array. And then we measure spike properties, extracellular spiking and bursting behavior of these organoids over the course of 30 minutes, and we do separate sets of recordings. And so this is extremely preliminary data, but just with a few organoids from control and these fused organoids, there appears to be, it's not significant, but there may be a trend towards an increase in spiking and bursting behavior when you look at those organoids coming from the CHD2 patients compared to the healthy controls. So that's potentially interesting and maybe a sign, at least there may be a difference in the way the circuits are being developed or maybe their network properties, and this is definitely something we want to follow up on to really identify what's the source of these differences. And of course, another way of investigating the physiology of the neurons is to measure intrinsic activity of the neurons. And so what Sona does is she takes these same fused organoid structures and she makes a slice from them, or she just directly puts a patch electrode into the organoids, sort of blindly, because at this point we don't really know what part of the organoid is where we are, and we're really just honestly just trying to find anything that's active. And so again, just from a couple of organoids between control and patients, there appears that there may be when you inject a current into these organoids, that perhaps the CHD2 organoid neurons fire higher numbers of action potentials at the same injected current. But again, this is extremely preliminary. Okay, so I have a very short talk today. Really, I feel that this work is very preliminary. We were very fortunate to have Vanessa get a grant from the LGS Foundation to allow us to start this work, because we had no idea what we were doing, and certainly this is such high-risk research that NIH would not fund at this very early stage. So first and foremost, we want to thank the LGS Foundation, foundations like this. Now with these systems in our lab, we can definitely do more studies with organoids, and we also are very excited to collaborate with other people who have other models, because we want to understand how this is consistent and translates across models. We recognize that these organoid models there's some major challenges and limitations. They are very variable. They don't have blood vessels, so they tend to have a highly necrotic core, and they lack those really important non-neural cell types that promote a functional maturation. And so at the same time where these technologies need to be optimized, but we can already begin to at least start interrogating some of these basic questions. And hopefully we will move one step closer to finding drugs and therapies for the patients. So with that, I want to thank Vanessa and Parul and all of the work that the people in the lab are doing. We also want to thank our collaborators, our UTSA Stem Cell Core, and also Alessandra Ranieri from the University of Siena, Italy, for the cells. And also we want to thank the other cores and the patients and their families for volunteering and consenting to be part of our project, and our financial support from NIH, from the American Epilepsy Society, and from LGS. Without their, again, I already mentioned the support, this work would not be possible. So thank you.